Hello, everyone. Welcome to the MIF++ seminar. It's a great pleasure to introduce Professor Manuel Muller, today from King's College, London, who will talk about spontaneous protein backbone rearrangements. Over to you, Manuel, please.
Thanks very much, Vitaly, for the introduction and for the invitation. I'm an interdisciplinary scientist. We work at the interface of chemistry and life sciences. So I'm quite used to giving talks to diverse audiences, typically ranging from maybe med schools to chemistry departments. But today is certainly a first in terms of presenting to a group of scientists at the interface of maths and material science. So I tried to adjust the contents slightly, but I'm a chemist at heart, so there's no escaping chemical structures, but I hope that's still an area of mutual interest. So my lab, as Vitaly mentioned, is interested in understanding how protein function can change over time. And this is both an interesting question in terms of understanding aging, but also trying to see whether there are opportunities that nature exploits to generate maturation systems. So as you know, proteins are the workhorses of biology. They carry out most tasks in living systems, ranging from motion in muscle movement to energy conversion, molecular recognition in antibodies, for example, in catalysis, and pretty much everything that nature does is to some degree dependent on proteins. But just like their macroscopic counterparts, these molecular machines suffer from wear and tear, and we're interested in understanding these processes. And to give you a bit more of a sense of the sort of the timescales that operate here, I've prepared a time graph with the ranges of times that chemists are interested in. And this is exciting because it's super broad. We get to think about timescales all the way from the jiggling of molecules and atoms all the way to geological timescales around the origin of life and these processes that sort of take billions of years to develop. And so as a chemical biologist, I want to zoom in a little bit on biomacromolecules and look at the hydrolytic stability of some of these molecules. So for a frame of reference on the bottom here, I'm just showing sort of timescales that make sense to a physicist, but maybe here are some more approachable timescales, especially on the larger timescale areas that are maybe a bit difficult to appreciate. So for those of you who are PhD students, this is about how long a PhD takes. This is about how long a master's takes. This is how your typical PhD viva will take, and my talk will be somewhere around here. If we look at the key biomacromolecules and the key bonds that hold them together and think about, well, how quickly do they hydrolyze under typical conditions, which sort of represent a boundary for maximum lifespans that living systems might reasonably have. At the lower end of the spectrum is RNA. That's the most labile molecule. It sticks around typically for years, and if you're pretty delicate at handling it, you can get your RNA molecules to survive for about an entire PhD, but anyone who's worked with RNA knows that that's actually much harder to accomplish than it sounds. DNA is much more stable than RNA, which makes sense given that it's the information storage polymer. So you want to have a very stable molecule. And so roughly the hydrolytic stability of this molecule dates back to timescales like the ascent of humans. So this is a really stable connection. Proteins are sort of intermediate stability timescale. They can stick around for about a century. And then probably the most stable biomacromolecule are carbohydrates. The acetal groups in carbohydrates are incredibly stable from a chemical point of view, and that makes them really suitable as building materials in biology and as energy storage materials. So let's zoom in a little bit into sort of the more biologically relevant timescale. So this is the lifetime of the average human nowadays, lifetime of a fly, and the doubling time of E. coli, sort of on the faster end of the spectrum. And let's have a think about how proteins fare in this timescale. So the average protein in our body will stick around for maybe about a week. At this point, then, it will get degraded and replenished by new protein biosynthesis to generate fresh proteins that will carry out the same task. Some proteins, however, are exceptionally long-lived. Histones, for example, and I'll talk a lot more about what histones are and why they're important, they can stick around with us for 100 days at least. And some proteins, such as I-lens crystalline, which plays a really important role in setting the refractive index of the eye lens, that protein is never replenished, so we're basically stuck with whatever we have at birth. And this is also a cause for why, during aging, many of us will get turbidity developing in the lens, because the molecular aging processes that take place on these proteins affect their ability to function. So in terms of the chemical processes that will occur to a protein over their lifetime and the lifetimes of us, is on the one hand, there's the hydrolysis, which is not typically a huge issue given how stable peptides are. Protein folding is very fast, with the slowest step in protein folding typically the flipping of proline from the cis to the trans conformation and vice versa. This happens on the time scale of minutes, but it's a reversible process, and so not usually thought as a very damaging thing to happen. However, there are some covalent changes that much more profoundly change the chemical makeup of protein that do occur during their lifetime. And the most prevalent of these is the spontaneous rearrangement of asparagine residues, here shown in the left, to isomerically linked isoaspartate residues, where the backbone has essentially hopped onto the side chain, generating this extended backbone, distinguishing it from the normal shorter backbone of proteins. So this process is initiated by the nucleophilic attack of a backbone amide nitrogen on the side chain amide, which then deamidates to generate this 5-membered cyclic intermediate. And this intermediate itself is unstable, and it can hydrolyze either this bond or that bond. If this bond hydrolyzes, you generate an aspartic acid. So you restore the typical backbone, but you've changed the side chain from an amide to an acid. So this is a formal deamidation process. But if this bond here hydrolyzes, then the backbone nitrogen hops onto the beta carboxylate, and you get this extended structure that I've mentioned. And so the timescales for this depend on local context of sequence and structure, and can be anywhere from days to years. And so this is a very biologically relevant timescale, so it's one that we need to understand. At the even more slow timescales, this process can also start with aspartic acid, where still the backbone nitrogen attacks the side chain of aspartic acid, but that's much slower under physiological conditions, and we're now sort of reaching timescales from years to centuries. So now we have this really interesting intersection between chemical processes that happen within the lifetime of proteins, and we really want to understand, do these processes negatively affect the structure and function of important biomacromolecule? So are there any functional consequences of molecular wear and tear? And here we define molecular wear and tear strictly just as this one specific chemical process, so we have a handle on it. And then a more long-term goal of my lab that I won't talk about today is, how do we detect this molecular aging or the occurrence of this isoaspartate in sort of a complex system? And so as a model system for answering this initial question, we have chosen histones and chromatin because, as I'm showing here, these are quite long-lived. But they're also biologically incredibly important because they play a key role in shaping our chromosomes. So each cell in our body has DNA that comprises, if you sort of array all the DNA molecule in a linear fashion, you get about 2 to 3 meters of DNA, which has to be compressed to a micrometer-sized compartment, the cell nucleus. And that's a challenge, because DNA is very negatively charged, and bringing all this negative charge into such a small space is electrostatically difficult. But also, this DNA has to be accessible, because our cells need to continuously read the information that's present in DNA. And not only do they have to read the whole thing, they also have to read it in a very cell type specific manner. So this compaction has to not only generate a spatially defined arrangement, it also has to be dynamic, where a brain cell, for example, can only read the genes that are important for brain cell function, whereas a heart cell, for example, would only need to be able to read heart cell genes. And a really important role in maintaining this architecture is played by a set of basic proteins called histones. Their names H2A, H2B, H3, and H4. These four proteins will together assemble into an octameric disk. That disk is very positively charged on the outside, and that provides an opportunity for negatively charged DNA to wrap around it. So you basically have a spool of proteins around which the DNA is wrapped, and that can compact the DNA, and then there's further compaction by some of these species sticking together. So in terms of nomenclature, this architecture is called a nucleosome. That is the basic repeating unit of chromatin, which comprises these histone proteins and the DNA that's wrapped around it. And if you look closely at these structures, you will see that from this relatively solid core, there are peptide tails that protrude. And these types of tails can act as antennas, and they orchestrate a lot of the interactions between machinery that reads, interprets, and repairs chromatin and the underlying sequences around. So these play a really important role. And given the long-lived nature of histones, it's not surprising that isoaspartate residues have been found in several of these histone isoforms. And we were particularly drawn to a modification that has been found at this position of histone H4. And this position to us is very interesting because it sits right at the interface between the globular core and these flexible tails. So they're essentially representing the connectivity between the signaling antenna and sort of the core where all the information is stored. And so we really want to know, how does molecular aging, which introduces these unusual amino acids at an important position in chromatin, how do these changes affect chromatin structure and function? So we don't know exactly the half-life of going from a young histone to what we would call an old histone, which has this isomeric amino acid, but we can estimate that this will take about 100 days. And in order to answer the question that we set out to answer, we can't just take this protein and let it sit on our bench for 100 days. That would be super convenient, but over these timescales, many things will happen, including the protein will aggregate. So we will never really know, we'll never be able to access a site-specifically aged protein. And this is where one of the unique specialties of my lab comes in, and also the reason why I'm in a chemistry department, is we can use chemical synthesis to generate these site-specifically aged proteins and then compare their structure and function to control proteins that are synthesized in their young form with a natural alpha linkage. And what I'll present now is the work of a former PhD student, Tianzi Zhang, who's now at the CRIC as a postdoc, and a postdoc, Luis Guerra. And so what Tianzi has done is she used a process called protein semisynthesis to generate these aged species. And this process is essentially a synthetic approach that takes the best of both worlds. We use chemical synthesis to make relatively short peptides into which we can introduce any chemically stable building block. And that includes these beta-linked aspartates, which we can introduce in a stepwise fashion. But where chemistry is not ideally suited is making long proteins. Proteins are relatively large molecules, and with each chemical step that you operate on, you'll get less and less of your final product. So we basically just synthesize the specialized part, but we outsource the production of the rest of the protein to E. coli, because E. coli is really good at producing proteins. That's how it lives. And we can instruct it to generate for us some of these protein fragments, and we can then use specialized chemistry to bring the synthetic and the recombinant fragments together to make the full-length protein, in this case, carrying this age-related lesion of an isoaspartate. And to enable this chemistry, what's really important, and probably the most chemical slide that I'll have in my whole talk, is that we need compatible ends between these proteins that allow the specific essentially gluing together of these two fragments. And this is done through thioester chemistry. So our synthetic fragment will have a C-terminal thioester that will react specifically with the N-terminal cysteine. So that is a natural proteinogenic amino acid, by the cysteine attacking the thioester to generate a new thioester that now covalently captures the synthetic peptide and the recombinant protein, albeit through this unnatural linkage. But fortunately for us, and this was developed 30 years ago, the species will undergo an energetically favorable shift where this entire peptide chain hops from the sulfur to the nitrogen to restore a native peptide bond, hence the name native chemical ligation. A specific adaptation we had to use in our case, there is no cysteine in this histone sequence, so we had to chemically remove the sulfur, which was incredibly important for the chemistry, but not present in the final product. But we can do that through radical chemistry to restore an alanine residue. So that was the synthetic design. I'll very quickly walk you through how we do this in practice. So we synthesize our peptides on a solid phase. So these are plastic beads that are reactive, onto which we can add, in our case, hydrazine, which represents the C-terminus of our peptide. And then through semi-automated peptide synthesis, which is done mostly by a robot, we can add our individual building blocks, chemically activate them, and one after the other install them on this plastic bead so that we generate the entire sequence of that synthetic peptide tail that we wanted to generate. We can then cleave this finished peptide off from the plastic support to generate the a soluble peptide, which we can purify using chromatography, and by mass spectrometry we can identify that we've made what we think we've made, and then we can chemically activate the C-terminus through oxidation chemistry that I won't go into detail to generate the thioester that is ready for this ligation chemistry that I mentioned earlier. I'll skip over some of the synthetic challenges, but I want to highlight, so in the first instance we made the young version of the peptide tail because that's easier to make, it's cheaper, and it's always good to do your methods development on something that's cheap. But we can, in the same fashion, make the old version of this tail by using a commercially available building block that's just slightly differently chemically arranged. We can incorporate that into the automated synthesis, and then using a couple more molecular detours, we can generate the same peptide, which differs from the previous one that I've mentioned, just in the configuration of this single residue. And so this would now represent here in green the old tail. And again, we can use purifications and mass spectrometry to ascertain that we've made the right thing.
Would it be okay to ask a question here?
Yeah, sure. Yeah, absolutely. Sorry, I can't really see. Oh,
on the previous slide.
Yeah.
Since we are not chemists,
Sure.
probably it's very simple for you. You mentioned you use mass spectrometry to understand whether well you got what you expected. So could you clarify when you mentioned a change in covalent bonds, for example? So how exactly you know? For example, do you know completely the atomic structure, but you don't crystallize with proteins, right? You do it differently.
Yeah, it's a very good question. So these peptides, we can't crystallize them. If we absolutely had to, we would use NMR, but typically it's accepted in the field that mass spectrometry is the best way of analyzing these peptides. What mass spectrometry will tell us is that the overall weight of the peptide is correct. And we know the sequence because we've basically instructed our robot to make it one piece at a time. Now, we won't be able to distinguish the old and the young version of these peptides because they have the same mass. They just have a slightly different configuration.
Mm-hmm.
So what we had to do there is they have different retention times by chromatography. That's serendipitous. They have slightly different physical chemical properties. And so we just kind of know which one's which because we've made them both. And we can show that they form distinct pieces. This has actually been a big problem during the synthesis, which I've completely glossed over. when we made the old version, we got a lot of contamination from the young version, so we had to adjust our process, changing how we do the deprotections, making it much more stepwise than we originally had. So that's an important consideration. The other thing you can do then is biochemical verification. There are means that I'll explain later with which you can identify whether you have an isoaspartate or an aspartate. And we've done that, but I don't have the data here.
Okay, thank you, Manuel.
No worries. Yeah, feel free to ask questions anytime. So then we took these synthetic peptides, ligated them with the bacterially produced protein fragment to generate the full-length species, desulfurized them, and again we use mass spectrometry to identify that we've got the full-length protein now. And in fact, we've done this for the young version on the left in blue, and for the old version on the right in green. Again, we can't distinguish by mass. They're isomers of each other, so they'll have the same mass, but we've labeled all the tubes so we know which one's which, and we can biochemically verify that the old is old and the young is young. So with the proteins in hand, we then incorporated them into chromatin templates. These are either individual nucleosomes or arrays of nucleosomes where we have 12 copies of these nucleosomes in a line in one sort of one DNA molecule. And with both young and old versions in hand, we could then begin asking questions of how does this molecular aging affect important aspects of chromatin function? And we've picked 3 specific questions that we wanted to ask because these are all important for chromatin function, but they're all involving this tail that we've chemically synthesized. And so we actually expected, or we hypothesized, that a change in this tail would actually negatively affect all of these parameters of chromatin. So the first thing we wanted to know is, does this aging affect how chromatin can compact? I've mentioned earlier that compaction is really important for fitting into the nucleus. And this is accomplished by one of these histone tails with its positively charged tail binding a negatively charged surface on a neighboring histone. And just through this electrostatic association, you bring the chromatin together and it becomes more compact. The easiest proxy to measure this with is just through a precipitation assay. The more compact the chromatin, the easier it is to precipitate it out. And we thought, again, this was going to be affected because the aspartic acid, which we've chemically aged, sits right between this flexible tail and the core sequence. And when we did the experiment, we basically got no discernible difference. So that means that despite this involvement, the overall ability of old chromatin to compact remains intact. So although our hypothesis was wrong, this is actually good news for biology, showing that our chromatin is reasonably robust against this form of aging. The next thing we wanted to know is, can chromatin still be remodeled effectively even once it's aged? And this was done in collaboration with Beat Viertz's lab at EPFL, and Jana Berlina did the experiments. Here again, these chromatin remodelers are molecular machines that will latch onto these antenna-like tails and then just spin around the nucleosomes that are attached to. And so you would expect, if you change the connectivity between where this molecule latches onto and where it does the work, you would get an effect. But the data suggested that actually the old chromatin, here shown in green, can still be very efficiently remodeled compared to the control young chromatin. One of the reviewers has pointed out, and one could argue this question, that there is a difference in the level of the plateau. We weren't prepared to really argue on the details, but I think the take-home message here is clear, that despite significant aging processes, remodeling can effectively take place. And so the last biochemical assay that we performed is around histone methylation. And you can imagine histone methylation is essentially the installation of a chemical bookmark into chromatin that signals to a cell this area of chromatin is important or not important for you. And so installing these bookmarks is incredibly important, and any mistakes in these processes can lead to cancer. So it's important that we understand them, and we were really curious how this aging-related changes in position 24 might change chemistry that goes on, or biochemistry that goes on at position 20, which is sort of an important biological signal. And this is done by two different proteins called set8. Set8 will install a first methyl group, and then a second protein comes in called SUF420H1. The name is not important, but it just adds a second methyl group in a stepwise fashion. So in a way, we had two shots to test and see whether aging affects this process because there are two separate proteins involved in the process. Again, structurally, we expect that there's some involvement because of the proximity between the aging modification and where the chemistry happens. And in the case of set8, this is just a loose connectivity. But in the case of the second enzyme, there's really a direct interaction between aspartic acid 24. So we expect this to be quite vulnerable to any changes. The experiment here we did is through mass spectrometry. We've measured the change in methylation once we mixed here young chromatin with both of the enzymes. And over time, we can see a diminished signal for the unmodified substrate and an increased signal here in orange for the first methylated species, which plateaus, and then a slow increase of the second methylation species. So that's on the young chromatin. If we take old chromatin, finally, for the first time, we see a significant difference. We can see much slower initial methylation, much more of the unmethylated species remains, and an almost complete abolishing of the second methylation step. And so the first step is slowed by about a factor of 3. The second step is slowed down by a factor of about 10. One of the reviewers actually suggested a really good experiment after this. What we've done here in our chemical approach is we've generated what some could call ridiculously old chromatin. This is almost like an unrealistically old model. What will happen in biology is you start with mostly young chromatin, and 1 or 2 of the components will age, but not all of them at the same time. And so to address this question or this criticism, what we've done is an experiment where we've generated chromatin that has substoichiometric levels of the old histones. So most of or half of the histones are young, half of them are old, and then we've measured the first methylation step and found that the slowing down of this methylation step is proportional to the number of old histones that we have. So that again suggests that the effect that we're generating is local, and this aging does not completely destroy chromatin, it just is a local damage. And so to conclude the first part of my talk on histone aging, what we've done is we've chemically synthesized old and young chromatin, and we found that chromatin is remarkably robust macroscopically against any sort of negative effects from this aging process, but there are some local vulnerabilities that arise from this aging process. And we think that the vulnerability actually correlates with the extent in which this specific residue is involved in the biochemical transaction. And we think that the macroscopic robustness is really important. It's an evolutionary pressure because it buys time for any sort of repair processes or even degradation processes that can kick out these old species before the entire cell or the entire chromatin sort of blows up due to aging. And so we've only looked at one specific process so far, and we think the time is ripe to study many of the other processes of molecular aging that might affect chromatin so that we get a much more systems level understanding of this aging process. And so in the second part of the talk, I want to switch gears a little bit. What I've mentioned earlier is essentially systems that are quite long-lived, where there's an evolutionary pressure to ensure that the proteins and the systems associated with it are robust enough to enable the associated organisms to live for a reasonably long time. But given this chemistry, there's also an evolutionary opportunity that nature might be able to exploit this process to access amino acids with really unusual structures and functions. This type of backbone connectivity is quite different to what you have in a normal protein. You have this extra methylene group, which introduces a kink in the otherwise extremely flexible bonds of the backbone, and that might provide an opportunity to generate new types of secondary structure elements. And I personally embrace the hypothesis that anything that's chemically possible, nature will have found a way to exploit it. And this is exactly the question we've wanted to ask. And as a model system, we used a bacterial enzyme called MurA. What is remarkable about this protein is that it contains stoichiometric amounts of isoaspartates in one of its hairpins. And we know this because of the extremely careful structural biology work done by the Schönbrunn lab about 25 years ago. And you can see, those of you who look at protein structures on a regular basis, this electron density map is perfectly fit by an isoaspartate residue here shown in black. And that enables a particularly tight hairpin in one of this protein's beta sheets. This protein is also quite important because it catalyzes the first committing step in bacterial cell wall biosynthesis. The chemical details of the reactions are not important, but in the long run, this chemical intermediate will be part of the bacterial peptidoglycan cell wall. And while bacteria have a cell wall, we don't. And so that provides an option for selectively inhibiting these. And indeed, there's an antibiotic called fosfomycin, which specifically inhibits MurA. So understanding this protein, which is an important drug target and a biochemically important species, is, I think, very important to do. If we look at the genes and the amino acid sequence, this isoaspartate is genetically encoded by an asparagine, which is followed by a glycine. And so this must be a post-translational conversion of the asparagine to a glycine. And whenever a biologist wants to ask whether a process is important, the first thing that they do is they try to see, is this process conserved? And what you can see here in this sequence alignment is that this structural feature is conserved among Enterobacteriaceae. So that's a subgroup of bacteria which include some environmental bacteria, which we have in us, but that can also be very infectious if injury arises, but also causative agents of cholera. So these are definitely important species. But there are other bacteria who have a very similar enzyme that generate entirely different structure. So it seems that this isoaspartate containing hairpin is one of several solutions for a topological problem that nature is trying to solve. And I think it's a particularly elegant solution because these hairpins are incredibly tight compared to the much longer hairpin sequences in these other bacterial species. It is worth noting, especially since some of you like looking at PDB structures and maybe correcting some of these records, the structure of Vibrio MurA is reported as containing an asparagine. But when we looked at the details, you can see that the asparagine here in yellow actually very poorly fits the electron density. And an isoAsp here on the right fits the electron density much better. And similarly, the originally fit asparagine residues, they violate the Ramachandran angles, and they have really distorted amide geometries. A stable amide would have a geometry of about 180 degrees with some fluctuations, but in some of these cases, these are 120, so they completely would have lost all hybridization and stabilization from the amide bond. So this to us was pretty good evidence to suggest that these isoaspartates actually are conserved in all these species. So to test whether protein species or protein samples that we produce in-house also would have these isoaspartate, we've resorted to this biochemical assay that I've alluded to before in answer to Vitali's question. There is a specific enzyme called protein isoaspartyl methyltransferase, which can repair this isoaspartate damage. And it's a methyltransferase that will just transfer a methyl group from adenosylmethionine onto the isoaspartyl carboxylate, which is an entry point to generate this succinimide intermediate, which can then hydrolyze again, stochastically in either of these positions. So it's quite an inefficient repair process, but it's one that we can exploit to check and see if we do see a methylation from this enzyme, we know we've had isoaspartate, and if we don't get any methylation, we didn't have any isoaspartate. And so we did that with our MurA sample, and we found almost no isoaspartate present if we do our assay under native conditions. And we were really surprised because there's this beautiful crystal structure that shows this protein has an isoaspartate, but for some reason we can't seem to methylate it with the enzyme. But then we thought, well, this residue is actually embedded in this tight structure. So probably what we'll have to do is denature the protein to make it accessible to the repair enzyme. And lo and behold, if we do that, we get an almost stoichiometric amount of methylation. So that means there was an isoaspartate, it's just not accessible to the repair enzyme, which also prevents a futile cycle in bacteria. And on the right, these are just control species to show that our assay is working. So we know that there is an isoaspartate present. We wanted to know how quickly this is formed. And so we tried to produce and purify this protein as quickly as we could, and then measure in vitro how long it takes for the isomerization to take place. And what we found that under native conditions, here shown in blue, the timescales of this process are just a few hours, about 5 hours, which is significantly faster than any previously measured isoaspartate formation, and also significantly faster than if we do this whole process under denaturing conditions, or with an isolated peptide that is not able to fold into the three-dimensional structure of MurA. And so this data clearly shows that this is an autocatalytic process. The three-dimensional structure of this protein accelerates its molecular maturation into an isoaspartate-containing species. And just to juxtapose that into the previous timescale, so this is really significantly faster than the expected range of 1 day, just based on the sequence. And because the protein accelerates this process, we thought it probably does it so for a reason. So this must have a positive effect on the structure or function of MurA. And so our first hint that this might have actually been true is when we just looked at how much protein we could generate from E. coli if we produced either a wild-type species, which will post-translationally form this isoaspartate, as compared to a mutant of the protein, where we mutate the asparagine, that's the genetically encoded residue at position 67, which cannot, under these timescales, form the isoaspartate. So this protein species is a constitutional isomer of the wild type that does not have the beta residue, but otherwise it's exactly the same protein. And we get about only half the amount of protein of the mutant, and also a significantly larger fraction of this protein is present in insoluble or soluble aggregates. So this suggests that this wild-type hairpin actually helps with producing, folding, and stabilizing the protein. The effect on stability is relatively small. We did chemical denaturation to measure this. The wild-type has a slightly higher midpoint of denaturation than the mutant, so it's measurable but not huge. And there seems to be an ever so slight effect on the activity, although this relative change is certainly small, and despite the error bars being quite tight, I would not confidently say that the wild type is more active than the mutant. This might be explained by very small changes in oligomeric state or folding. And so we wanted to know in more detail how this hairpin might stabilize the protein. So we teamed up with Argyris Politis group, who is an expert in structural mass spectrometry, and they measured hydrogen deuterium exchange mass spectrometry, which is a way in which you could measure the dynamics and the accessibility of parts of a protein. And so without going into the technical details, what their analysis showed that only the red parts of the proteins have significantly different dynamics between the wild type and the mutant. So in the mutant protein, these red sections are much more floppy than in the wild type. And more precisely, we were able to narrow this down both to the actual hairpin and the structures underneath the hairpin that were stabilized by an isoaspartate. So essentially what we think is happening is that this tight hairpin actually acts like a lid on the secondary structure element. And if you mutate it, you just get this opening and closing of the lid, which destabilizes the structure, exposes hydrophobic surfaces, which might lead to aggregation of the protein over some time. So I hope I was able to convince you that these spontaneous chemical processes can have two very distinct phases. They can, on the one hand, represent molecular wear and tear, where the responsibility of biology is to protect itself from negative effects of this wear and tear. And in the context of chromatin, nature has done a pretty good job of doing so. But also that the same chemical processes can be exploited to access interesting and different structural element, which can stabilize proteins. And we're also exploring the possibility that nature might use this process to build molecular timers that have different functions in the young and in their old state. But I unfortunately don't have time to talk about that today. What I want to do in the very last section of this talk is essentially a call to arms for people who are interested in protein structure and function to start looking more at the polypeptide backbone. And I say this because the backbone of a protein actually has half of every protein's mass. It's just in the backbone. It's incredibly important for the 3D structure. You will know that backbone hydrogen bonding is important for helix formation, for hairpin formation, beta sheet formation. But it's also, the backbone has very different reactivity depending on the sequence context and the structural context. And so that presents an evolutionary opportunity to tune the reactivity of the backbone to maybe generate some of these maturation processes that I've mentioned earlier, but many other options. The sequence context also provides a potential for molecular recognition by proteins that might edit the backbone. And so there's a huge area of unscoped biology that might come from being able to edit the backbone. And so far, we've been really limited to serendipitous discovery. We've been at the whim of incredibly careful structural biologists who have noticed these very subtle differences of isomers not being fit perfectly by electron density maps. And there's very few tools that allow systems level discoveries for these modifications. And that's something that my lab is really passionate about. In the last couple of slides are data not from my lab, but for some examples of the types of backbone modifications that do occur that have been found in the PDB. So for those of you who like traveling through the PDB and finding interesting structures and connectivities, this might be something interesting to look at. So nature has found ways to edit pretty much all aspects of the backbone. You will see changes to C alpha. You can find changes to the oxygen atom of the backbone and to the nitrogen of the backbone amide. The first one I've already mentioned with great detail, that MurA can have this isoaspartate, but also one of the most abundant proteins in many cells, one of the ribosomal proteins, appears to have an isoaspartate in its midst. The function of this entirely unknown. Modifications at C alpha include isomerizations to D amino acids, which is again something that can happen during aging, but on very slow timescale. But there are also peptide venoms that have D amino acids. Some proteins will have dihydroamino acids that have a planar trajectory from the side chain and the backbone. And there are some quaternary substituted amino acids. And some examples of two of these are actually in an enzyme called methyl coenzyme M reductase, which does really interesting chemistry in methanogens and also represents a really abundant protein in these species. And the electron density map again shows that there's a dihydroaspartic acid in one position and an alpha methylated glutamine, so a quaternary center, which is very unusual in proteins. Modifications of the amide nitrogen include succinimide formation or a redox active 5-membered ring formation, for example. And an example here is from the beautiful work of the Balaram lab, who have shown that these cyclic structures can provide a high degree of stability in thermostable enzymes. And lastly, the oxygen atom can be replaced. There's an example for proteins that have thioamides in there, where the oxygen is kicked out, the sulfur is installed. And the only way you'll see that is the electron density cloud is just a little bit bigger than of an oxygen. So again, that requires really good data and really careful analysis. One example for here, for the third time today, is methyl coenzyme M reductase, but also, again, a ribosomal protein seems to have a thioamide in one of its originally peptide bonds. And the last example that I want to give is one that most biologists will have seen a lot, but often don't think about how it's actually generated. That is the beautiful green fluorophore of green fluorescent protein is actually generated through a series of backbone condensations and oxidations, and it embodies all three of the different types of modifications that I've mentioned to generate this conjugated system that gives green fluorescent protein its characteristic green glow. And with that, I want to close. I want to thank all my students and postdocs for doing the work. I've only talked about the work of Luis and Tianze for the most part. Unfortunately, I didn't have time to go into the details of what everyone else does. I want to thank our collaborators in Beat's lab and Regulus Politis lab, also Jane Allison, who's done some computational work on MurA that I haven't talked about. The funders for funding this, and thanks to all of you for your kind attention, and I'd be very happy to take questions.
Thank you very much, Manuel. Let us please thank Manuel for the insightful talk. Yes, so physically, virtually. Let me now stop the recording.